We're investigating various methods for the rapid display and manipulation of three-dimensional medical images. This is a series of two-dimensional slices which have been obtained through a standard computed tomography examination. These slices may be stacked to form a three-dimensional object. These three-dimensional objects may be displayed and manipulated using a variety of tools which we have developed. We have built an interactive physician's workstation prototype using a special-purpose graphics processor designed and built in the GRASP lab called the Voxel processor prototype. Workstations of this type have applications in clinical diagnosis, surgical planning, radiation therapy planning, and other clinical areas. The Voxel processor prototype allows the real-time display and manipulation of medical images. Interactive facilities include three-dimensional rotation via a trackball, slicing and multiplanar reformatting, interactive segmentation through grayscale thresholding, and the display of dynamically changing objects. After the images are acquired, two-dimensional slices are displayed on a monitor so that the user may select a three-dimensional region of interest for display. The user selects the region by positioning the cursor via a graphics tablet. The region of interest is then loaded to the Voxel processor. Here the user has selected a portion of the cervical spine, and thresholds away soft tissue, exposing only the bone. A trackball allows the user to interactively position the object so that it may be seen from any orientation. The Voxel processor displays and manipulates small volume elements called voxels. Special digital hardware is used to produce realistic images of medical objects on a conventional video monitor. The prototype generates approximately 16 frames per second. Slice planes provide the important ability to view the interior of objects, as seen with this spinal section. Full-resolution CT data of the eye and the orbit is seen here. As we reslice the eye at an arbitrary angle, anatomical structures such as the lens, optic nerve, orbital muscles, and orbit become clearly visible. Windowing allows for clear visualization of the orbit. Here we see a number of two-dimensional slices of a brain as obtained from a PET scanner. When this data is loaded to the Voxel processor, the three-dimensional structure is revealed quite vividly. Windowing shows the exact location and concentration of the radiopharmaceutical tracer.